Thank you, Emmanuel. So it's a great pleasure to introduce Meredith Cenzer to this audience. Meredith did her PhD in UC Davis, was really young on insect-plant interactions. She finished her PhD in 2016. And since then, she moved on a more theoretical postdoc with Lethan McGonigal in Florida State University on the evolution of dispersal. And then moved to something a bit more different, University of North Carolina at Chapel Hill with Daniel Matute on the plasticity mating behavior of Drosophila. At the same time, well, during the same period, she was a visiting scholar at Columbia University. In 2019, she works at the Ecology and Evolution Department of the University of Chicago. Meredith's work is largely focused, I think it's fair to say it's largely focused on the interaction between selection and gene flow in shaping local adaptation and maladaptation, and how plasticity can modulate these processes. She works mostly on insect herbivore interactions, using the soapberry bug as the major insect of her studies. Her work is also relevant to biological invasions, since one of the host plants of the soapberry bug she studies is an invasive species. I actually first came to know indirectly, not directly, Meredith, when I handled a paper she submitted to the American Naturalist when I was an editor of that journal. And I was immediately impressed by the quality and the depth of the paper. And I was even more impressed by the quality and the depth of the answers that Meredith provided to all the comments that were raised by reviewers, the associate editor, and myself. And it sort of naturally followed from all this, at least for me, that paper won the Student Paper Award of the American Naturalist that year. I will stop here. Meredith, we're all looking forward very much to listening to your talk, which I'm sure would be very exciting and very interesting to this audience.
Thank you. Thank you for that introduction, and thank you for inviting me here today. I'm really looking forward to sharing some of my work on maladaptation in a changing world. And so I'm just going to jump right into it. So where I'm going to start today, make sure I can progress forward. Yeah, where I'm going to start today. So here's my roadmap for my talk. So first I'm going to talk about kind of what I mean when I say maladaptation. As evolutionary biologists, we often think about processes as being adaptive. And so when we're thinking about the converse of that, it's useful to set it up at the beginning, kind of, well, what exactly do we mean? How might we detect maladaptation in nature? Then I'll introduce you to my system, the red-shouldered soapberry bug, and talk about the history of that system and the kind of shifts in that system from a local adaptation story to more of a maladaptation situation. Then I'll spend the bulk of my talk really talking about maladaptive plasticity, both in morphology and in behavior. And then I'll talk a little about the most recent work I've been doing in my lab on dispersal in soapberry bugs. And then I'll wrap it up with a little bit of context in thinking about how these single species cases can impact the entirety of the local community. And so I'll start with what I mean when I say maladaptation. So I really like this definition from Crespi in 2000, who defines maladaptation as prevalence in a population of a strategy, that is a form of a phenotype, that does not lead to the highest relative fitness of the strategies in the allowed set. So basically, if we have a population where most individuals are expressing a phenotype that is not a good fit to that environment. But of course, that's a relative measure. So we really need to think about how we're defining this allowed set. So if it's not possible for this species ever to express a certain type of phenotype, should that really count as maladaptation? And I would argue that no, it should not. So we want to look at what the allowed set is. And we can do that by either looking within a population. So if some of our individuals have a high fitness phenotype, but most of them do not, we know that phenotype is possible. But if it is rare, we might call that maladaptation. We can look at variation across space. So if some of our populations are expressing a phenotype that would be a good fit somewhere else, but doesn't occur in the right location. If we have changes across time, so if our population historically had higher fitness phenotypes than it does today, and we're seeing a movement away from adaptation, away from that fitness peak over time. Or we might look across environments, so at plasticity. And what this might look like, and I'm going to give some examples of this further into the talk, is if we have an organism and when it experiences a given environment, it then expresses a plastic phenotype that is not well suited to that environment. Even though perhaps in a different environment, it could express a phenotype that would be well suited to that first environment. So we live in a world undergoing huge amounts of environmental change, largely caused by human activity, that have created a lot of opportunities for organisms to experience environments that are well outside of the range of environments they have an evolutionary history with. And so when we take a genotype and we put it in an environment it has no history with, we really don't know what it's going to do. And so this is really a great opportunity to see maladaptation in action because of that absence of evolutionary history. And in many cases, what's interesting to me about these human modified environments is that they occur in a patchwork on the landscape, where we have some locations and some populations experiencing habitats that still look like or similar to their ancestral habitat, while other populations are experiencing highly modified environments. And so we can see the whole dynamic play out of colonization of a new environment, what happens right away, what happens over time, so seeing that process of adaptation potentially, which we have seen in many systems, and then how that then might feed back into what's happening in those more ancestral looking environments. Because none of this happens in a vacuum. We can have individuals moving between these habitats. And as we see human modified environments becoming increasingly prevalent on the landscape, we might see a shifting balance from having all of those populations persist to seeing one or the other dominate. So now I'm going to tell you a bit about my specific system with the red-shouldered soapberry bug and patterns of adaptation and maladaptation over time in the system. So here they are. These are red-shouldered soapberry bugs. They are seed feeders that specialize on the seeds of a subset of plants in the Sapondaceae or the soapberry family. And the seeds of these host plants are defended in a variety of ways that are very important from the perspective of the soapberry bug. So some of them will defend their seeds with a large inflated seed pod like this one. And soapberry bugs, excuse me, like all true bugs, feed through an elongated beak, which is basically like a pointy straw with two little saws at the end that they use to drill into the seed. And so because they don't have mandibles or hands, they can't open a closed seed pod. And so if they want to feed on the seed inside of a closed pod, they need to have a beak that is long enough for their body to remain outside the pod and their beak to reach the seed inside. And so in this system, we have seen repeatedly across different species of soapberry bugs, beak length evolving to match the local seed pod size. I work on this system primarily in Florida, where there are two host plants. There's a native host plant pictured here on the bottom, balloon vine, with locations here in pink circles. And this host plant defends its seeds with a large inflated pod that looks like a little balloon, which is where it gets its name. And both soapberry bugs and balloon vines are native to Florida. So these have a very long-standing evolutionary history together. And I'll sometimes refer to this as the ancestral host plant. And in the mid-1900s, golden rain tree, with the tree pictured here on top and inset of the seed pod itself, this host plant was introduced to Florida as an ornamental from Taiwan in the mid-1900s. And I'm showing locations here in blue triangles. Unlike balloon vine, this seed pod is both very flattened and is open for the majority of the development of the seed. So while the balloon vine seed pod presents a complete barrier to feeding for soapberry bugs whose beaks aren't long enough, the golden rain tree seed pod is wide open. And so you don't need to have a long beak to feed on this host. And in fact, if you have a long beak, it really will slow you down. So soapberry bugs colonized golden rain tree after its introduction. And by the 1980s, local adaptation was first documented in this system. But I like to mention a little bit about the community living on the native host plant, because none of these species occurs in a vacuum. So while on the golden rain tree, the soapberry bug is the only thing I've ever seen eating the seeds or the foliage. Well, they don't eat the foliage. I don't see anything eating the foliage, but the only thing I see eating the seeds. While on the native host plant, there's a much more complex community. So there are at least 2 other species of soapberry bugs in the same genus, these 2 up top that are smaller than the red-shouldered soapberry bug. There's at least 1, maybe 2 hair streaks whose caterpillars develop on the seeds of this host plant. And there is 1 galling wasp, and I will say this is not the same species because there are no pictures of that one, but there are galling wasps that develop on the seeds as well. And then there are foliar feeders, such as this invasive iguana. So I want to put that out there just so you have some context to think about what's happening on this native host plant beyond soapberry bugs. So for the soapberry bugs themselves, local adaptation was first documented in the 80s. And here I'm showing histograms of beak length on the top half of this plot, and then pod size on the flipped upside down histograms. And what you can see is that for beak lengths for females feeding on balloon vine, they had much longer beaks here in pink than bugs collected from golden rain tree in blue. And that there was very little overlap between these, which I'm showing here in this purple cross-hatched region. And we can see that beak lengths at this time were diverging in the direction we expect based on our prediction that beak lengths should evolve to match local seed pod size. And while I'm not going to talk about the other phenotypes of local adaptation today, this was also true for juvenile survival, development time, and for egg size. And in the same period, this beak length difference was documented as really just being genetic. So Scott Carroll and colleagues did cross-rearing experiments where they raised individuals from these 2 genetic backgrounds on the seeds of both host plants. And they found that consistently, those from the balloon vine genetic background had longer beak lengths, regardless of what host they developed on, than bugs from golden rain tree. So I returned to this system. I've been working there since 2013 and have since found a really huge shift away from this pattern of local adaptation. So what I'm showing here are aggregated beak length measures from across 2013 to 2020. I'll show them split up in just a second by year. And what you can see is, first, that while these used to be really quite distinct, they are now completely overlapping with the entire distribution of golden raintree beak lengths covered by the distribution of balloon vine beak lengths. So they're no longer distinct. What you also might notice is that there has been a shift, particularly in beak lengths for bugs on the native host. So there's been a marked reduction in the average beak length for bugs from the native host, while there has been very little shift in the beak length for bugs on the introduced golden raintree. And so this is the same pattern we see in other phenotypes, so in survival and development time, suggesting that what we're seeing here is increased gene flow from our populations living on golden raintree to our populations living on balloon vine, swamping out that local adaptation. And so if we look at this across time, we have measures here from the late 80s and early 90s from Scott Carroll and colleagues showing really striking local adaptation between the first two populations and then a set of 5 populations. And then here I'm showing data that I've collected over the past 8 years. And we can see that while there are some years where it looks like local adaptation peaks out, so say, for example, in 2013, this was the most local adaptive looking year I've seen. But we also see years where there's really no difference at all, and even years where that pattern is flipped. And so we've clearly moved away from local adaptation into a pattern of broad overlap. And if we break this up across space, something else interesting pops out. So here I'm moving from north on the top to south at the bottom, with the top 4 populations collected from golden raintree and the bottom 4 from balloon vine. And we can see here, I'm showing the mean here as this solid bar, although you notice there is some bimodality, which I'd be happy to talk about later. And there's really not much change in the mean, at least, moving through these top 3 populations. But then something happens when we hit the sympatric zone here, where we suddenly see this very striking difference between our bugs collected from golden raintree and those from balloon vine. And then when we move out of the sympatric zone down into the keys, where it's just the native host, we see this return to more intermediate phenotype. And so when we see this pattern of stronger differentiation in sympatry, many of us might think of character displacement, where our populations are being driven apart by niche differentiation, so selection to specialize on different hosts. But what's most striking about this is that it's happening in the wrong direction. So our bugs on golden raintree ideally should have short beaks, while our bugs on balloon vine ideally should have long beaks. And so we see this stronger differentiation in sympatry, but in a maladaptive direction. So why might that be happening? Well, first I'm going to talk, so next I'm going to talk about maladaptive plasticity, which I think is one of the key drivers of this. And so in this piece, I wanted to ask, how does the developmental host influence adult morphology? So historically, there was no effect of the rearing environment, or the rearing host at least, on adult beak length. Is this still the case, since we see this pattern happening where we've seen a total shift in this system? So is this still the case, and does it look adaptive? So for this experiment, I did a cross-rearing experiment where I took bugs from populations across Florida, and I reared their offspring on the host, either the native host or seeds of the introduced host. And what I found was this very striking pattern where, one, we see that there are still genetic differences between populations collected from these two host plants. So from the balloon vine genetic background, overall, they do still have longer beaks than bugs from golden rain tree populations. So this is that same old pattern of local adaptation, but it's being totally masked by plasticity in beak length. So for bugs who develop on balloon vine, which is where we need a long beak if we're a bug, they develop shorter beaks than bugs who develop on golden rain tree. And so in the field, these two effects perfectly mask each other, where bugs from the golden rain tree genetic background who develop on golden rain tree have beak lengths that are indistinguishable from bugs from the balloon vine genetic background who develop on balloon vine. So why is this happening? Why do we see this maladaptive plasticity? If you're capable of expressing a long beak, you should ideally do that when you're in the place where a long beak would be beneficial, which would be balloon vine. Well, I'm very fortunate to work in a system with a rich history of studies. And so it turns out that Scott Carroll and colleagues did an experiment, a set of experiments in the 90s, looking at what happens when you hybridize those ecotypes. And so they did this experiment with bugs collected again in the early 90s, where they looked at the effect of golden rain tree ancestry percentage on the beak length bugs express. So what I'm showing here on the x-axis is golden rain tree associated ancestry. Oops, sorry. With those pure ecotypes here, this being 100% balloon vine ancestry, and over here 100% golden rain tree ancestry. And then in the middle, we have 50% ancestry from each, so hybrids, and then our two back crosses. And what we can see is that while in the 90s, there was no effect of developmental plasticity on beak length for our pure ancestry ecotypes, there was a very strong effect of plasticity in hybrids and back crosses. So individuals with mixed ancestry were expressing this maladaptive plasticity historically, even though pure ecotypes weren't. And so if we're seeing increased gene flow in the field today, it may be that this plasticity is the result of a high prevalence of mixed ancestry individuals in the field, where they're expressing this maladaptive plasticity that used to be restricted to artificial crosses. So if this is the case, we would expect to see higher plasticity where gene flow is highest and lower plasticity where gene flow is lowest. And so we would expect to see plasticity being strong in the sympatric zone and then becoming weaker as we move away from the sympatric zone. And indeed, that is what we see. So in the zone of sympatry and near the zone of sympatry, which is where most of the native host plant occurs, we see that there's a much larger difference between beak lengths for individuals raised on either host plant. And then as we move away from the zone of sympatry, we see that return to no plastic effects on beak length. And so the way that I think about this is that while there are still genes that allow for long beaks happening here close to the sympatric zone and in the keys, because these individuals have lost the ability to effectively use the native host plant as a resource, they're not able to express those long beak genes. And so we've seen that the developmental host can have a really profound effect on adult morphology, but that is highly variable between populations. And so I wanted to then take this knowledge that we have now about plasticity as a result of the developmental host and take this into a behavioral context. So from the perspective of an individual soapberry bug, or really most animals, mate choice is one of the most important decisions that an individual makes as an adult because it determines their reproductive success and whether their genes will successfully persist going forward. So we know that the developmental environment has a strong effect on adult phenotype. So we might hypothesize that this will also carry over into how attractive those individuals are as mates. So I then wanted to ask, how does developmental environment influence mate choice? And again, does it look adaptive? So given that we've seen these increased rates of gene flow, mate choice could have a contributing role there, where if our individuals who develop on golden rain tree actually look more attractive because they're better at using that host plant, we might expect them to be more attractive as mates and therefore be more effective agents of gene flow. In contrast, we might expect that because balloon vine is the ancestral host, perhaps there's been some persistence of that host really being necessary to develop cues that look attractive for mate choice. So I did a series of experiments for this. So for this experiment, I'm going to talk about the effects of the developmental environment on mate choice. And I did this with a cross-rearing experiment. So I took bugs from populations on golden rain tree and populations on balloon vine, and I reared their offspring on the seeds of both of those host plants, as pictured here on the right. And then when they reached adulthood, I put them into preference trials. We had one female and two males, one from each rearing host treatment. And for this experiment, I wanted to look specifically at the effects of plasticity, not at choosing between genetic backgrounds. So I held population constant. So for every trio I'm going to talk about, they all come from the same population, but different rearing environments. And so once they reached adulthood, I put them into preference trials that look like this. And soapberry bugs have prolonged mate guarding. So you'll often see them out in nature looking like this. So this is our female, and she is in copula with one of the males, while the other male looks longingly on. And they can remain in copula for hours or up to days. So these were very long preference trials. So they ran for 48 hours. And in the first hour, I scanned them continuously to look at latency to mate, who the female mated with first, active rejections of male mating attempts by females. And then I checked them again every 2 hours, so for 10 total observation periods over those 2 days, and looked at who the female was mating with at each repeated check and the number of attempts males made to mate with the female. So it's a measure of male preference. And then once the preference trials were over, I allowed the female to lay eggs for 8 additional days as a measure of reproduction. And then everyone went into the freezer and we measured their morphology after the experiment was over. So what did we find? Well, first I want to show this because I'm always really happy when experimental results are repeatable. So first I looked at morphology. And here I'm showing the beak length effects of developmental environment again. So happily, they still look the same as they did when I did the previous experiment with bugs, I believe, that were collected in 2014. And so these are bugs from 2019, and they show the same pattern, which is very encouraging. So we're still seeing that balloon vine bugs have longer beaks overall, but that, again, we're seeing they're masked by that maladaptive plasticity. However, I did not see any effects of morphology directly on mating success, so I'm not going to talk more about morphology in this experiment. So if we're looking at mate choice, I'm going to break up these results into the female perspective, so who was she actually mating with between these two males, and the male perspective, so how frequently males attempted to mate with with the female in their treatment. And so to orient you to this plot, excuse me, here I'm showing this on the x-axis, the genetic background, so bugs from balloon vine trios here on the left, and bugs from our golden rain tree trios will be here on the right. And on the y-axis, I'm showing the fraction of observations where the female was mating with the golden rain tree treatment male. So if it's up here, that means that group of females only ever mated with the male from the golden rain tree treatment. If we're down here, they only ever mated with the male from the balloon vine treatment. And there'll be a dotted line across the middle showing the no preference line. And I will also split this up based on where the female herself developed. So whether there's an impact of the female developmental experience on who she ends up mating with. And we can see that there is, first, a really clear effective genetic background. So our populations, our trios from balloon vine, show a very strong preference for mating with males who developed on balloon vine. And this is particularly true if the female herself developed on balloon vine. Where in contrast, our golden rain tree trios show no preference at all, so they're equally likely to mate with a male who developed on either host plant. So to summarize that piece, so females from the balloon vine genetic background have a strong preference for males from the balloon vine treatment, where females from the golden rain tree background express no preference. So what about from the male perspective? So to orient you again here, I'm showing the genetic backgrounds split up in each panel, with the male developmental host here on the x-axis, and again, the colors will denote which host the female developed on. And I'm showing the number of mating attempts each male made during that 10-minute period. And remember that these are no choice assays from the perspective of the male. So there's only one female in with the male. So his options are to attempt to mate with that female, to not attempt to mate at all, or to attempt to mate with the other male, which happens pretty often, but I unfortunately did not record. So first, looking at the balloon vine genetic background, we can see that males are about 3 times as likely to court a balloon vine treatment female. So there is a very strong preference here, with males being more active in courtship if the female in their trio developed on the ancestral host. And if we look at the golden rain tree background, we see first that this preference is still there. So across the board, all males prefer to court females who developed on the ancestral host, but that there's an interaction here where the difference in courtship behavior is weaker for the balloon vine, or for the golden rain tree genetic background. So to summarize that, males from the balloon vine genetic background, similar to females from that genetic background, have strong preference for mates who developed on balloon vine. And our males from the golden rain tree genetic background have a weaker preference, although still in the same direction. So how does this impact adult fitness? Oh yeah, I'm just jumping right to it. So are they making an adaptive choice? Well, no, they are not. So here I'm showing from the perspective of the female, the number of eggs each female produced based on the amount of the time that she was mating with a male from the golden rain tree treatment. So excuse me. So the more time a female spent with a male who was from the golden rain tree treatment, the more eggs she laid. So this is the opposite of the preference that anyone is expressing. So an adaptive choice here would be to mate with the male who's going to help you produce more eggs. And in this case, we're seeing females preferring to mate with males who do not do that. So we're seeing them making a maladaptive choice here. So what about from the male perspective? Maybe females who develop on balloon vine lay more eggs than those who develop on golden rain tree. Well, spoiler alert, they do not. So across the board, females who develop on golden rain tree lay more eggs than females who develop on balloon vine. And there's also a strong genetic background effect. So bugs from our golden rain tree genetic background are much more fecund than bugs from our balloon vine genetic background. And so to put all of this together, while we have these strong preferences for both males and females from the balloon vine genetic background for mates from the balloon vine treatment, this is not reflective of which mates will give them the highest reproductive output. While in our golden rain tree genetic background, preferences tend to be weak or absent, but still not in an adaptive direction. And so we might conjecture that this is either, so I have two kind of hypotheses about why this might be. One, it could be that the ancestral preference is for individuals who develop on balloon vine. So there may be some compounds from balloon vine that are typically expressed and make mate attractive, but those are absent on golden rain tree. And so after colonizing golden rain tree, those populations began not to use those cues because they weren't present. And so they've lost some of that preference. Conversely, it could be that the ancestral state is to have weak or no preference, and that a preference for a balloon vine specific cues has evolved more recently in the balloon vine genetic background as a response to avoiding maladaptive gene flow. But I will say that based on the negative effects of these choices on reproductive output, regardless of the origin, these are likely to be really to have negative effects on our populations in the Keys because they're already struggling to maintain population sizes that are robust to an influx of gene flow from outside of those islands. So this is not likely to help them regain local adaptation in the face of ongoing gene flow. And so I then want to tell you just a little bit about an area that I've been working on a lot. So when we're thinking about gene flow between populations, really the first step that has to happen before gene flow takes place, before gene flow takes place, is dispersal. So individuals have to be able to move between these populations to carry their genes from one place to another. Historically in this system, these were considered to be essentially allopatric. So it was thought that they were mostly too far apart for dispersal to be an issue. Now, clearly that's not the case because we've seen this pattern of gene flow between populations, and we've seen that it's enough to swamp out a lot of that local adaptation. But we don't really know how far individuals are capable of dispersing, who those dispersers are, and what the impacts of that might be. So I'm just going to give you a little preview of what I'm working on here without a lot of results. So I'll show you a couple of preliminary things on dispersal. So what we've set up in my lab, and I want to credit Anastasia Burnett, my wonderful research technician with this. Let's see if I can get it to play. There we go. So this is a flight mill that we've built to measure insect flight. And what you can see, we've got 8 different chambers here with individual soapberry bugs attached to magnets with magnetic paint that fly in a circle attached to a spinning arm. And each time that arm makes a rotation, it breaks an infrared sensor beam and is recorded as a dip in voltage here. And so we can measure not only individual propensity to fly, but also individual flight speed and distance. So we have a lot, many hours of recordings for this that we've been sifting through to understand what these patterns look like. And the first thing that really jumps out at us that I'll just briefly show you here. Well, first, we were not expecting soapberry bugs to be that good at flying. So even though we know there's been gene flow between these populations, when you see them, typically they're very clumsy flyers, and you wouldn't expect them to be very successful at long distance dispersal. But when we put them on these flight mills, we really learned that they can go for a long time. So we've seen bouts of flight for 15 kilometers in a single bout, where they will keep flying continuously for more than 10 hours. So they are really capable of very impressive dispersal events. And I'm really excited to see what we can do with that distance data. But first we looked at flight propensity. And one of the things that was really striking here is that males fly twice as much as females. So when we're thinking about who those early dispersers are, who the agents of gene flow are, it is really likely to be males who are arriving at the other site. And for males, we saw this strong sexual dimorphism in flight. And for males, this is driven really by wing morphology. So males who have longer wings for their body size are more likely to fly. But this is not true for females. And something that's also interesting about this, while a lot of morphology in general is strongly plastic based on the developmental host, this particular metric is not. So wing to body ratio is very consistent. It's very conserved regardless of the developmental environment, suggesting that this is something that males at least are really maintaining. So they're investing in this over other morphology. For females, in contrast, it's really driven by their mass, which is perhaps unsurprising because they have to carry it around, which is strongly influenced by their reproductive status. So a reproductive female is very unlikely to be the one dispersing. So they'll be dispersing either before becoming reproductive or after bout of reproduction has ended. And I'm going to leave it there for dispersal, although I'm excited to have more on that soon. So I want to wrap it up by putting this in the context of what this might mean for the rest of the Native community. So this system might seem unusual, but as human modified environments are increasingly colonized and adopted by Native species, we've seen now many species where they have adapted to use a human modified environment. And that pattern of gene flow back into that Native system, really changing the face of the entire species, I think is unlikely to be rare, especially as these modified environments become increasingly common. And so that can manifest in a lot of different ways that we might not expect. And in this case, we've seen some really unusual plasticity, or what seems unusual, but maybe isn't, plasticity in behavior. But now that this species has changed, what is that going to mean for all of the organisms that are interacting with it. And so in this case, we've got this complex community of other organisms that feed on the native host. And so we might start seeing shifts from the soapberry bug being the dominant seed feeder on this species to one of these other seed feeders becoming more common. And anecdotally, it does seem that the silver-banded hairstreak over here, this lovely green butterfly, has become more common since the 80s and 90s when surveys were done. And so this might have ecological impacts and also potentially have cascading impacts on the selection pressures that individuals, or not individuals, but that populations are experiencing when feeding on this host. So one thing that I've been thinking about a lot recently is how this might impact selection on seed pod size in the native host. So if soapberry bugs no longer have beak lengths typically that are long enough to reach inside a large pod, we might see a shift away from selection for large pod size on this host plant. And to wrap it up with this slide that I showed at the beginning, so we've seen this breakdown of local adaptation in beak length. We've also seen potentially what looks like a decrease in pod size on this host plant. And so I will suggest that this could be a response of a second species to this increase in gene flow and loss of local adaptation. And so with that, I'd like to thank my undergraduate research assistant, Anna Silberg, my fabulous research technician, Anastasia Burnett, the First Nations of Florida on whose unceded land much of this collecting was done, and the Florida Park Service for permission to collect in state parks. And with that, I will take any questions.
Thanks, Meredith. So people can still go on and type their questions on the Q&R tab. We have one at least at the moment, but there will be more coming. And while we wait, I would like to share exactly the same anecdote as Yanis, in fact, because I was an associate editor, not editor, at Evolution, I think for your first PhD paper where you were the single author and mostly had the same experience, and I remember it in the same way as Yanis. So you did this twice at least, which is
thank you.
Enough to note. So we have a question by Thomas Lenormand. So, so let's see if I can turn on his microphone. So Thomas typed his question, but we're going to ask it to you directly. Thomas, can you speak?
Yeah, I can speak. So perhaps you hear me. Yes, very interesting. Thank you. I was wondering, I have the impression that the distribution of big sizes is very variable, is very within population, even excluding those hybridization effects. And so I was wondering, and also because there is such a strong plastic response, whether there was, I don't know, for instance, year-to-year variation, of pod sizes or something that would explain why there is such a variation, or plant to plant or something. I don't know.
Yeah, so there certainly is variation in pod size, and I have some measures of that I haven't analyzed yet, so I won't speak to that too much. There's also a lot of variation in body size. And so when I'm showing those raw histograms, that's not accounting for that variation, although I account for it in analyses. So in some of these where you see like a bimodal distribution, I don't know if that's kind of what you're thinking about, and I can hop back up to that. That some of that is because of variation. Ooh, no, I would like to escape this.
You mean you've got juveniles or something?
So it's all adult females in these histograms. But there are different, I didn't talk about this, but there are different wing morphs that really create bimodality in body size between adults. So, which is most likely what's kind of creating this large spread, so that there are smaller individuals who have shorter beaks just because of allometry, and then larger individuals who have this kind of classic long wing morph. But yes, so there's a two morph situation where everyone who has this kind of short flightless morph has a smaller body size and correspondingly smaller everything, including smaller beaks.
And I suppose you did try to remove the effect of body size to see if the relative big size and
Yes.
see whether and is it a different pattern with that?
It's the same pattern, yeah. So when you account for body size, the pattern remains. Yeah. Okay,
thanks.
All right, then we have a question by Vincenzo Ostra. Vincenzo, you want to ask your question? Are you still around? Yes. I don't know if his microphone works. Otherwise, we can read it.
Hi, can you hear me?
Yes.
Okay. Yes, I was just wondering whether you know anything about the developmental mechanisms underlying this plasticity and the hybrids, kind of in this cascade of sensing the host plant until like they develop this longer beak. Do you know anything about that? And sort of, I'm just curious how they became plastic when they were previously not.
Yeah, I think that's a really great question that I would love to know the answer to. So in terms of like the developmental genetics of it, I do not know. So there is a lab that is working on developmental genetics in the system, who has previously worked primarily on wing morphology, so rather than beak length. So we don't really know what it is. Yeah, but I would love to. Great question.
Thanks.
Yeah, so I would have sort of a follow-up to that. Even if you don't know the mechanism, I was curious by the fact that the more like the F1s were more plastic than the native population, and then the backcross a little less. So this seems like because I mean, from what I understood, and there were some old papers by Gillespie and Torelli on this, that the more heterozygosity there is, the more you expect some kind of buffering between plasticity of different backgrounds. And I mean, I would expect that there's less plasticity in more, I don't know. I mean, this is just based on this model, basically. So I don't know if you've thought about this. This is a bit striking and surprising to me to find more plasticity in these heterozygotes, or more heterozygous than the other. These F1s, at least if I understood well, this is what you found, right?
Yeah.
So found in this older paper, and then what you found in this sympatry versus allopatry, right?
Right. So, like, why is there more plasticity here in the hybrids and back crosses?
Yeah, I mean, or I don't know if you have an answer, or you've been thinking about this, but
yeah. So the way I explain it to myself is thinking about, there's both what's happening with the genes directly impacting beak length, and then there are the genes relating to actually letting them metabolize the host at all. And so if you're in an environment that's really stressful and restrictive because you don't have the genes to actually use it, it doesn't matter if you have like genes that would give you a really long beak, because you can't express them anyway because you're too stressed out by the environment you're in. And so I guess I'm thinking of it like, first you have, and this is totally just like what I think would be happening, that you have some pathway that lets you actually get the nutrients you need. And you have to get through that before it even matters, like before you can even express the genes for a long beak length. And so here they have this kind of co-evolved gene complex where they can deal with this host plant and they have the genes for a long beak, but then you get in here and I would think much more, it's easier to kind of break that ability to use the native host effectively than it is to break long beak expression. That's
You're thinking of this in terms of hybrid breakdown and incompatibilities, something like that. Leads to maladaptive plasticity. Okay, thanks. Let's see. Oh, Sarah, so we have a question by Sergio Gonzalez. So let's see if I can turn on the microphone. Sergio, can you want to ask your question?
Yes. Hi. This is actually really interesting, and yeah, I've been wondering also a little bit about this, but trying to make the link that with what Luis Miguel was saying, that maybe this F1 had like a higher heterozygosity, and that would allow it to have sort of more plasticity. If we link, for example, the phenotype ability to express to genetic diversity, and then after that, in next generations, there is sort of a local selection process that reduces this heterozygosity and therefore also the plasticity. Do you think that that could be happening?
Perhaps, yeah. So I could see that. I don't know any more specifically than that. Yeah, so it's like, but there could be gene combinations that are getting purged like before they reach adulthood. Is that kind of what you're?
Yeah, more or less. Yeah.
Yeah, that certainly could be happening. Yeah.
Okay, interesting. Thank you.
All right.
Thank you.
All right, so let's keep to Vincian's question now. Vincian. You can talk now if you want.
Okay, thank you. And thank you for your great talk. I was wondering, maybe you said it and I missed it. Do you know if there is a threshold between the number of laid eggs and the survival of the offspring in this species? I mean, have you looked at the viability of all the eggs that you count?
Yeah, so that's a great question. I did not in this study, and that's definitely an important caveat. Like that it could be that you lay more eggs and a higher proportion of them are inviable. So yeah, so I don't know for this particular case. Certainly they're not like obviously inviable, or we wouldn't count them. So sometimes they lay clearly inviable eggs, but because we didn't look at hatch rates, we can't say for sure. In previous experiments that I've done, hatch rates have been pretty consistent, but of course there could be impacts, you know, on viability or even on adult fitness or, you know, fit to the local environment. So there are a lot of other measures of offspring fitness that I did not include. So definitely worth thinking about. Question.
Okay, thank you.
All right, anyone has more question can raise their hands also if they want, their virtual hand that is. Perhaps Yanis had a few comments because he cannot ask them in the Q&R. Yanis, do you have any more things to add on this about the talk? No?
Sorry, can you?
Yes. Yeah.
Can you repeat what you said, because my connection just?
Oh yes, sorry. I was wondering because I was wondering if you had a few comments or questions. You
yeah, actually, I had a minor question. They're both related to the relationships you showed between the flight probability and traits in males and females. So the first question with respect to this is that you show the relationship between wing length in the males and the flight probability, and the relationship between, yeah, that's right, wing to body ratio. Okay. And in the females, the relationship is driven by body mass. So I was wondering whether, because a priori wing length and body mass are likely to be correlated in the females, I was wondering whether you had corrected for this when looking at the.
Yeah.
Yeah.
So absolutely, you know, when we look at the models with both of those factors in them, there's actually a weaker relationship between those than you might expect, because wing to body ratio is already itself as a measure corrected for body size. It doesn't have that. It doesn't have a correlation with body size on its own, while mass, of course, does have a relationship. But because females can really expand or reduce the size of their abdomens as they're reproductive, that relationship is much weaker with mass. One other thing we've done to look more directly at this, because mass itself is plastic, right, over the lifespan of an individual, we did multiple trials for each individual. And so we could also look at the effects of changes in mass between trials on whether or not individuals flew or how effectively they flew. And so I don't have those data here, but those show the same pattern, while of course that individual has maintained the same wing morphology across that period.
Okay, and perhaps more interesting, does this flight probability depend on either their genetic background or their rearing environment?
So these trials were done with field collected individuals. So we have not yet done an experiment where we controlled the rearing environment and then looked at flight. So I don't have an answer for that yet, but it is high on the list of things we want to look at. And we have not found strong effects of genetic background on flight probability. It does look like it impacts flight distance, but we're not done with those analyses yet. So waiting for more. Yeah. And of course, we'd like to control for rearing environment when we ask that question.
Could there be an indirect effect of the genetic background on flight probability through its effect on either the female mass or the male wing to body ratio, if these differ with respect to the rearing horse?
Yeah, absolutely. I think that is definitely possible. So the wing to body ratios are higher for golden raintree associated populations from field collected individuals across the board, and for cross-reared individuals from other studies. So so you would expect there to be a higher flight propensity for bugs from golden raintree because they have these relatively longer wings. In the field collected bugs that we tested, we don't see that. So it's sort of well, it's not really clear, I think, at this point, because the morphology is there, but we haven't seen the effect. So remains to be seen.
Okay, thank you very much.
All right, so I think we're going to finish here. I want to thank Meredith again for this great talk that combined evolutionary ecology with a nice natural history component, I think. So next week, there won't be a seminar because it's the weekend de l'Ascension. And then the next seminar will be the 21st and will be on COVID, I think, by Mircea Soponea. So looking forward to seeing you then. Bye-bye.
Thank you.
Thank you. Bye.